Welcome back, class. Today's lecture is lecture 3, Microbial Nutrition. In this lecture, we're going to take a look at the nutritional requirements of microorganisms. We're going to look at the classification of microorganisms based on nutritional types. We're going to look briefly at how nutrients are required by microbes, how microbes are cultured in the laboratory, and the different types of culture media that's used. Researchers have analysed the composition of microbial cells, and they've shown that over 95% of cell dry weight is made up of only a few major elements, as you can see here: carbon, oxygen, hydrogen, N, S, P, K, magnesium and iron. These are known as the macro elements because they're required in large amounts. The first 6 of these macro elements are components of carbohydrates, lipids, proteins and nucleic acids. The remaining 4 macro elements exist predominantly as cations in the cell, and they play a variety of different roles. So for example, the potassium ion is required for the activity of a number of enzymes. The calcium cation contributes to the heat resistance of endospores, among various other roles. The magnesium ion serves as a cofactor for many enzymes. It also complexes with ATP, and it's been found to stabilise ribosomes and cell membranes. Fe2 plus and Fe3 plus are part of cytochromes and also cofactors for enzymes, and they're also required by electron carrying proteins. In addition, all microorganisms require microelements in small amounts. So these microelements are needed by most cells. However, in the lab, because cells require such small amounts of these microelements, that normally contaminants from water, glassware and regular media components are actually adequate for growth. So it doesn't need to be added to the growth media. Microelements are normally a part of enzymes and cofactors, and they aid in catalysis of enzyme reactions and also in the maintenance of protein structure. So for example, the zinc cation is present at the active site of some enzymes, but it's also involved in the association of enzyme regulatory and catalytic subunits. The manganese cation aids most enzyme reactions. Molybdenum is required for nitrogen fixation, and the cobalt cation is an important component of vitamin B12. Now, all organisms have an absolute requirement for carbon, hydrogen, oxygen, and a source of electrons. Carbon, of course, is needed for the skeletons or backbones of almost all organic molecules from which organisms are built. And of course, those of you who have studied organic chemistry in living systems will also appreciate the importance of hydrogen and oxygen in organic molecules. Electrons, of course, are also essential. The movement of these electrons through various electron transport chains is essential for providing energy for the organisms and also in the reduction of molecules during biosynthesis. So for example, the reduction of carbon dioxide to form organic molecules such as sugars occurs during photosynthesis. Now, these requirements for carbon, hydrogen and oxygen are often satisfied together, because molecules that serve as carbon sources often also contribute hydrogen and oxygen. So for instance, heterotrophs that use organic molecules as carbon sources can also obtain hydrogen, oxygen and electrons from the same molecules. And indeed, the more reduced the organic carbon source is, that is, the more electrons it carries, the higher its energy content. And therefore lipids have a higher energy content than carbohydrates. However, there is one carbon source, carbon dioxide, which supplies only carbon and oxygen. It can't be used as a source of hydrogen, electrons, or energy. And this is because carbon dioxide is the most oxidized form of carbon. It lacks hydrogen and it's unable to donate electrons during your typical redox reactions. So organisms that use carbon dioxide as their sole or principal source of carbon are called autotrophs. And they have to obtain their energy and electrons from other sources. So for example, here we're talking about plants, algae, and photosynthetic bacteria. Now these organisms use water as a source of electrons and they release oxygen in the process. They have the amazing ability to split water molecules. So because the need for carbon, energy and electrons is so important, biologists use specific terms to define how these requirements are fulfilled, so that we can actually classify any living organism according to nutritional type. So we already can imagine that microorganisms can be classified as autotrophs or heterotrophs, just like all other organisms. Of course, autotrophs use carbon dioxide as their carbon source, as you can see here listed under carbon sources, and heterotrophs use reduced preformed organic molecules, which they obtain from other organisms. Our second classification here is according to energy source. There's only two available here. Phototrophs will use light as their energy source, and chemotrophs will derive their energy from the oxidation of organic or inorganic compounds. Under the subheading electron sources, again, there are two different types: lithotrophs, otherwise known as rock eaters. These microorganisms use reduced inorganic substances as their electron source, for example, sulfur, hydrogen, iron, methane, and so on. Organotrophs will obtain their electrons from organic molecules. So now looking at these three different subheadings, we can apply this to classifying any living organism according to nutritional type. That is, according to where they obtain carbon, energy and electrons. So let's start with classification of a human or a horse or even a fish according to nutritional type. And we start with the energy source. So if we just focus on the human and classify a human according to energy source, you can see that we would start with chemotroph. So let's start the name with the word chemo. Now let's move on to electron source. A human's lithotrophs or organotrophs. They obviously obtain electrons from organic molecules, so humans are classified as organotrophs. So far in the name we have chemo, organo. Now let's move on to carbon sources for the third and final part of the classification. Humans are not autotrophs, they're obviously heterotrophs, so the word heterotroph becomes the third part of the name. So according to nutritional type, a human is classified as a chemo, organo, heterotroph. Now let's use this same system to classify a plant or algae. Again, we start with energy sources. Plant or algae are phototrophs. So photo becomes the first part of the name. Moving on to electron sources, plants and algae are lithotrophs. So we incorporate the word litho into the name. So far we have the name photolitho, and then the third part of the name becomes autotroph. So plants and algae, according to nutritional type, are regarded as photolitho autotrophs. But despite the huge metabolic diversity seen in microorganisms, most of them can be placed into one of 5 nutritional classes that you can see here on the left, based on their primary sources of carbon, energy, and electrons. So the nutritional classes listed here are photolithoautotrophy, photoorganoheterotrophy, chemolithoautotrophs, chemolithoheterotrophs, and chemoorganoheterotrophs, with the first 2 classes being the most common. And it's probably worth noting that although a particular species generally belongs to one of these classes, some of them actually show quite a lot of metabolic flexibility, and they can alter their metabolic patterns in response to environmental changes. In other words, they can switch from one class to the other. For example, many of the purple non-sulfur bacteria act as photoorganoheterotrophs in the absence of oxygen, but at normal oxygen levels they can oxidize organic molecules and function as chemoorganoheterotrophs. So this gives them a definite advantage when environmental conditions change. In order to grow, a microorganism must also be able to incorporate large quantities of nitrogen, phosphorus, and sulfur. These elements are needed for the synthesis of important molecules, so for example for amino acids and nucleic acids. Nitrogen can be supplied in numerous ways. We'll come back to that on the next slide. Phosphorus is usually obtained as inorganic phosphate, and sulfur as sulfate. Let's look at nitrogen a bit more closely. So nitrogen is needed for synthesis of amino acids, nucleic acids, some carbohydrates, some lipids, enzyme cofactors, and also other molecules. Most microorganisms can use the nitrogen present in amino acids. Others can incorporate ammonia directly through the action of enzymes such as glutamate dehydrogenase or glutamate synthetase, and certain other enzymes. Most of the photo and chemotrophs can reduce nitrate to ammonia and then incorporate the ammonia. And this is a process known as assimilatory nitrate reduction. Some bacteria, so for example the photosynthetic cyanobacteria and also soil bacteria belonging to the genus Rhizobium, can assimilate atmospheric gaseous nitrogen, and they do this by reducing it to ammonium, and this process is called nitrogen fixation. Phosphorus is present in nucleic acids, phospholipids, nucleotides such as ATP, several cofactors, some proteins, and other cell components. Almost all microorganisms use inorganic phosphate as their phosphorus source and incorporate that directly. Keep in mind that low phosphate levels actually limit microbial growth in many aquatic environments. Some microorganisms, for example E. coli, can use both organic phosphate, for example hexose 6-phosphate, as well as inorganic phosphate. Sulfur. Sulfur is required for the synthesis of substances such as the amino acids cysteine and methionine. It's also required for the synthesis of some carbohydrates, biotin and thiamine. Most microorganisms use sulfate as their source of sulfur. Some microorganisms have the enzymes and all the biochemical pathways needed to synthesize all cell components just by using minerals and sources of energy: carbon, nitrogen, phosphate and sulfate. Other microorganisms lack one or more of the enzymes needed to manufacture essential cell components, so these must be supplied by the environment. Organic compounds that are essential cell components or the precursors to essential cell components that the cell cannot synthesize are referred to as growth factors. There are three major classes of growth factors, as you can see listed here: amino acids, which are needed for protein synthesis; purines and pyrimidines, which are needed for the synthesis of nucleic acids; and vitamins. Vitamins are small organic molecules that usually function as enzyme cofactors, and they're needed in only very small amounts to sustain growth. For your interest, here are some examples of common vitamins required, their functions, and the microorganism that requires each of these vitamins. Understanding growth factor requirements of microorganisms actually has important practical application. Those microorganisms that are able to synthesize large quantities of a particular vitamin can be used to manufacture these compounds for human use. Several water-soluble and fat-soluble vitamins are produced partly or completely using industrial fermentations. Here are some examples. Riboflavin is produced by Clostridium and Candida. Coenzyme A is produced by Brevibacterium. Vitamin B12 is produced, for example, by Streptomyces and Pseudomonas. Vitamin C is produced, for example, by Erwinia and Corynebacterium. Beta-carotene by Dunaliella, and vitamin D by Saccharomyces. The first step in nutrient use by microorganisms is uptake of the nutrients by the microbial cell. Microbes often live in nutrient-poor environments, so they must be able to transport from dilute solutions into the cell against a concentration gradient. So microorganisms make use of several different transport mechanisms. The most important of these are facilitated diffusion, active transport, and group translocation. However, a few substances such as glycerol, water, carbon dioxide and oxygen can move across membranes and cell walls by simple passive diffusion. But this process requires a fairly large concentration gradient from high to low concentration in order for adequate uptake by the cell. Larger molecules, ions and polar substances must move into the cell by other means. The rate of diffusion can be greatly increased by specific carrier proteins embedded in the plasma membrane, and this is called facilitated diffusion. However, it seems to be much more prominent in eukaryotic cells than it is in prokaryotic cells. And in eukaryotes, it seems to be used to transport a variety of sugars and amino acids into cells. Now, active transport, of course, is used to move substances against their concentration gradient. And this is particularly important because microorganisms often inhabit environments that are characterized by very dilute nutrient sources. Active transport uses metabolic energy, usually in the form of ATP. Now, another type of active transport mechanism called group translocation chemically modifies the molecule as it's brought into the cell. The best known system is called the phosphoenol pyruvate sugar phosphotransferase system. better known as the PTS system. And this is observed in many different species of bacteria. It transports a variety of sugars into the microbial cell while phosphorylating them using PEP, which stands for phosphoenol pyruvate, as the phosphate donor. For example, E. coli can take up glucose, fructose, mannitol, sucrose, N-acetylglucosamine, and various other carbohydrates using this system. Besides their role in transport, these PTS proteins can also bind chemical attractants towards which the bacterial cell moves by the process of chemotaxis. The study of microorganisms absolutely depends on the ability to grow and maintain microbes in the lab. And this is possible only if the appropriate culture media are available. A culture medium is a solid or liquid preparation used to grow, transport or store microorganisms. And in order to be effective, it must contain all the nutrients required by the organism for growth. Culture media are classified often on the basis of several parameters. One, the chemical constituents from which they're made, their physical nature and their function. So here we have the types of media defined by these three parameters. So first, according to physical nature, media may be liquid, semi-solid or solid. According to chemical composition, nutrient media may be defined or complex. And according to functional type, nutrient media may be supportive, enriched, selective or differential. A medium in which all the chemical components and their concentrations are known is referred to as a defined or synthetic medium. It can be made up in a liquid form known as broth or solidified by an agent called agar. And we'll come back to agar in just a moment. Defined media are often used to culture autotrophs, such as our photosynthetic cyanobacteria, and you can see an example on this slide called BG11. So this tells you exactly how to make up this particular medium to culture cyanobacteria. Another example shown here is medium used to culture E. coli. Defined media are often used in research as it's often desirable to know what the experimental microorganism is metabolizing. Interesting to note that not all defined media are as simple as these two examples shown here, but may be constructed from dozens of different components. Complex media contains some ingredients of unknown composition and or concentration. Complex media is particularly useful and very widely used because a single complex medium may be sufficient to completely meet the nutritional requirements of many different species of microorganisms. So for example, the first one listed here, nutrient broth, is one that you'll be using in your prac classes, and it's also very widely used in microbiology labs. Complex media is also required because often the specific requirements of particular microorganisms are unknown, and thus a defined medium cannot be made. Let's look at some of the media components found in complex media. Complex media contains undefined components like peptones. Peptones are protein hydrosylates prepared by partially digesting meat, casein, soya meal, gelatin and other protein sources. Peptones serve as sources of carbon, energy and nitrogen. Extracts such as beef and yeast extracts are aqueous extracts of beef and brewer's yeast. Beef extract contains amino acids, peptides, nucleotides, organic acids, vitamins, and minerals. Yeast extract is an excellent source of B vitamins as well as nitrogen and carbon compounds. Now, solidified media are particularly important, and they're needed to isolate different microorganisms from each other in order to establish pure cultures. We'll come back to the concept of pure cultures later on. The liquid media is solidified with the addition of 1.5% agar, although that might range from 1 to 2%. Agar is a sulfated polymer composed mainly of D-galactose, L-galactose, and D-glucuronic acid, and it's usually extracted from the cell walls of red algae. Agar is well suited as a solidifying agent for several reasons. One reason is that agar melts at about 90 degrees Celsius, but once it's melted, it doesn't harden until it reaches about 45 degrees, which means that after it's been melted and of course sterilised in the process, it can be cooled down enough to handle and to add substances such as antibiotics and so on, which would be destroyed at higher temperatures. And it can be handled in order to pour agar plates. Agar is also an excellent hardening agent because most microorganisms can't degrade it. Supportive or general purpose media supports the growth of many different species of microorganisms. One example is tryptic soy agar. Enriched media is also general purpose media, but blood and other special nutrients may be added to encourage the growth of fussy microorganisms. A couple of examples are shown here. So at the top we have blood agar, and growing on the blood agar is bacteria from the human throat. And at the bottom we have what's called a chocolate agar. And growing on this agar plate is the very fastidious Neisseria gonorrhoeae. The colour of the chocolate agar results from heating of red blood cells to lyse them before adding to the medium. And finally, selective media, which favours the growth of some microorganisms and inhibits the growth of others. One example is McConkey agar, otherwise known as Mac agar, and this type of medium selects for gram-negative bacteria and inhibits the growth of gram-positive bacteria. Another example of selective media is media that contains nutrients that only a particular type of microorganism can use. So for example, a medium that contains only cellulose as a carbon and energy source is quite effective in the isolation of cellulose digesting bacteria. Now let's look at two examples of nutrient media and their mechanisms of action. Starting at the top, blood agar. Blood agar supports the growth of many fastidious bacteria. These can be differentiated based on their ability to produce hemolysins. These are proteins that lyse red blood cells. Have a look at the agar plate, the blood agar plate rather, at the bottom left of screen. Hemolysis appears as a clear zone around the bacterial colony in the case of beta hemolysis, or as a greenish halo around the bacterial colony. This is known as alpha hemolysis. The clear or greenish halo around the bacteria depends on whether they completely lyse the red blood cells, or they simply modify the iron within the hemoglobin of the blood cells. Our next example is the third one in this table, McConkey agar or Mac agar. And you can see an example of bacteria growing on Mac agar at the bottom right of the screen. The selected components in Mac agar are bile salts and crystal violet, which inhibit the growth of gram-positive bacteria but promote the growth of gram-negative bacteria. The presence of lactose and neutral red, which is a pH indicator, allows the differentiation of gram-negative bacteria based on the products released when they use lactose as a carbon and energy source. Those bacteria that can ferment lactose as their carbon and energy source will release acidic waste products into the surrounding media, and this drop in pH is shown as red colony growth, and this is shown by the neutral red pH indicator. And this makes these bacterial colonies easily distinguished from the colonies of non-fermenting bacteria. So what we've just looked at are two different types of differential media. This media is used to distinguish between different groups of microorganisms based on their biological characteristics. So for example, blood agar will distinguish between hemolytic and non-hemolytic bacteria. McConkey agar will distinguish between lactose fermenters versus non-fermenters. Now, in natural habitats, microorganisms, of course, grow in complex mixed populations with many different species. And this presents a problem for microbiologists because a single type of microorganism can't be studied adequately in a mixed culture. What one needs is a pure culture. This is a population of cells arising from a single cell. And we need a pure culture so that we can characterize an individual species. Pure cultures are so important that the development of this technique by the German bacteriologist Robert Koch transformed the field of microbiology. Within 20 years after development of the technique, most bacterial pathogens responsible for human diseases had been isolated. There are several ways to produce pure cultures: the spread plate technique, the streak plate, and the pour plate technique. We'll be learning the spread plate and streak plate techniques in prac class, as these two are so commonly used. If a mixture of bacterial cells is spread out on a nutrient agar plate at very low density, every cell should grow into a completely separate colony, which is a macroscopic visible growth or cluster of bacteria on a solid medium. And you can see the various colonies here on this nutrient agar plate. Because each colony arises from a single cell, each colony represents a pure culture. The spread plate technique is an easy way to achieve this, assuming that a dilute mixture of cells is applied. But because we don't know how concentrated the cells are in the original culture, we can prepare dilutions and plate out a small volume of each dilution to try to achieve these well isolated colonies. If too concentrated, the colonies will all merge together, and this is referred to as a lawn of bacteria or confluent growth. Each colony is often referred to as a colony forming unit or CFU. We'll be looking more at this in our prac classes. So the successful technique really depends on the spatial separation of single microbial cells. Here we can follow the preparation of a spread plate. In one, a small sample of the bacterial culture is pipetted onto the centre of a nutrient agar plate. In two, a glass spreader is placed into a beaker of ethanol, and it's then briefly flame sterilised. The spreader is then allowed to cool down. In four, the bacterial culture is evenly spread over the agar surface with the sterilised spreader. The nutrient agar plate is then incubated at the appropriate temperature. Here we have an excellent example of a spread plate, a successful spread plate. Many of the colonies are well isolated from each other, so colonies can be easily counted for the purpose of enumeration of cells in the original culture. We can also very easily distinguish the different species of bacteria growing on this plate. Now let's look at the streak plate technique, which is also used to obtain pure colonies. The microbial mixture is transferred to the edge of an agar plate with what's called an inoculating loop, or in some cases a swab can be used. And then the mixture is streaked out over the surface of the nutrient agar in one of several different patterns. So let's look at step number one, where the first sector of the plate is streaked. Then the inoculating loop is sterilised, and the inoculum for the second sector is obtained from the first sector. A similar process is followed for streaking the third sector in step number 4, except that the inoculum is from the second sector. So this is essentially a dilution process, and eventually very few microbial cells will remain on the loop, and single cells will drop from it as it's rubbed along the agar surface. And these single cells will develop into separate colonies, otherwise known as pure cultures. Colony development can also aid a microbiologist in identifying microbes, because individual species often form colonies of characteristic size and appearance. Here we have variations in colony morphology seen with the naked eye. The general form of the colony shown in the first row, and the shape of the edge of the colony or margin shown in the third row can be determined by looking down at the top of the colony. Then, viewed from the side, we can actually determine the nature of the elevation of the colony as the plate is held at eye level. Elevation is shown in the second row. In the photos to the left and centre of screen, we have examples of commonly observed colony morphologies. In the photo to the right of screen, you can see how colony morphology varies dramatically depending on the medium on which the bacteria are growing. These snowflake-like colonies were formed by the soil bacterium Bacillus subtilis growing on nutrient-poor agar. The bacteria apparently behave cooperatively when confronted with poor growth conditions, and often the result is this intricate structure that resembles the pattern seen in non-living systems. The growth of colonies on agar surfaces has been fairly well studied. Generally, the most rapid growth occurs at the colony edge. Growth is slower in the centre of the colony, and cell autolysis takes place in the older central portions of the colonies. This is especially noticeable with yeast colonies. These differences in growth are due to gradients of oxygen, nutrients, and toxic waste products within the actual colony. The colony centre is much thicker than the edge, so oxygen and nutrients don't readily diffuse into the centre. Toxic metabolic waste products cannot be quickly eliminated, and therefore growth in the centre of the colony is slowed down or stopped altogether. In nature, microorganisms often grow on surfaces in what are called biofilms. These are slime-encased aggregations of different species of microorganisms. It's still not clear how characteristic colony patterns develop. Nutrient diffusion and availability, bacterial chemotaxis, and the presence of liquid on the surface all appear to play some role in the formation of patterns. Cell-to-cell communication is also thought to be important, but still a lot of work needs to be done to understand the formation of bacterial colonies and biofilms.